me wait a couple minutes. Sorry. Okay, I guess we should get started. It's my pleasure today to introduce this research seminar. It's based on CARB Research Seminar Contract 13-311. And the name of that contract was Co-Exposure to Ultrafine PM and Ozone, Pulmonary C Fiber, and Platelet Activation. Our speakers today will be Dr. Fern Tablin and Edward Schlegel from UC Davis. My name is Pat Wong, and along with Lori Miyasato, we are the contract managers for this project. Deborah Dressler was the original contract manager and integral to getting the funding for this project, so hopefully she's watching us right now. Now, just a number of announcements. For those of you online, thanks for joining us for today's seminar. Questions for the speaker can be sent to sierrarm@calepa.ca.gov. Additional information about the speakers can be found at the seminar link you see on the screen. Just a couple housekeeping announcements, or required housekeeping announcements for the attendees here. Please look around now and identify 2 exits closest to you. In some cases, an exit may be behind you. In the event of a fire alarm, we are required to evacuate the room immediately. Please take your valuables with you and do not use the elevators. Evacuees will exit down the stairways and possibly to a relocation site across the street. If you cannot use the stairs, you will be directed to a protective vestibule inside a stairwell. Should we have to relocate outside the building, please obey all traffic signals and exercise caution crossing the street. Okay, just one more announcement that's not on the screen. The speakers have accept our invitation for lunch after the seminar. So if you're interested in joining the speakers for lunch, please come to the seminar room, the Sierra room, about 5 to 10 minutes after the end of the seminar. Now, just a little bit of the project overview. We basically live in a multi-pollutant atmosphere, but almost all our health analysis and standard setting are done for single pollutants, and this is by design. We want to know the health effects for each pollutant. But then the question does occur, what happens when you have more than one pollutant with each other? Are they additive? Are they synergistic? Or are they even antagonistic? And we wanted with this project to kind of get the answers for 2 of our major criteria pollutants, particulate matter and ozone. Now, the methodology that the researchers used was they had mature hypertensive rats that were exposed to ozone and or ultrafine PM for 6 hours. The reason they chose this animal model is that mature individuals with cardiovascular disease are normally the most sensitive to ozone and PM. During the exposure period and the recovery period afterwards, cardiophysiology was assessed. And then after the recovery period, lung, heart, and blood samples were taken out and then analyzed for a variety of endpoints that they will discuss today. Now, why did we fund this project? Well, first of all, like I mentioned, we wanted to look at the combined effects of a multi-pollutant exposure and see whether or not the effects were additive, synergistic, or antagonistic. We probably would not use the study for standard setting, but this study can lead to future studies and add the biological plausibility for future multi-pollutant health effects. And this may impact future research directions and also our standards evaluations. Now, our speakers today, the primary investigator is Dr. Fern Tablin. She has a Bachelor of Arts degree in history, degree in veterinary medicine, and a PhD in anatomy from the University of Pennsylvania. She's currently a professor of anatomy, physiology, and cell biology at the School of Veterinary Medicine at UC Davis. Her expertise is in platelet physiology with multiple papers focusing on platelet physiology of domestic animals and animal models in health and disease. Her co-investigator is Dr. Edward Schlegel. He has a Bachelor of Science in Animal Physiology, a Master of Arts in Physical Education, and a PhD in Physiology from UC Davis. He's currently a professor in physiology and chair of the Department of Anatomy, Physiology, and Cell Biology in the School of Veterinary Medicine. His expertise is in how environmental factors affect human health and he has multiple publications examining factors that contribute to the exposure response of ozone in humans. And I guess with this introductions, I'd like to turn this over to Fern.
Thank you, Pat. It's a pleasure to be here, and we are delighted to present this work to you. We thought we'd give you the punchline, in essence, before the end. So what we said is synergistic effects of ultrafine particulate matter and ozone in adult animals with underlying cardiovascular disease. And really what we would like you to think about is how air pollution by inhalation affects the lung, and potentially, as it does that, these particles can make it into the circulation, can affect the autonomic nervous system of the heart, and both of those things can affect the heart and also the blood. So we have a lot of things to be able to talk about today. So I'm going to take just a second and give you our rationale and some of the background to think about. As Pat said, ambient air pollution is a major contributor to global disease burden. It's a major contributor to disease burden in the state of California. And epidemiologic studies show a positive association between exposure to fine, ultrafine, PM, and ozone, and adverse responses in people with cardiovascular disease. Acute exposure has been linked to increased cardiovascular events and mortality, and that's something that people really hadn't thought about for a long time. The presumption was that it required a chronic exposure. One thing we do know is that high incidence of exposure-related adverse health effects are present in mature populations, and in particular, these mature populations with underlying cardiovascular disease. These folks are at much greater risk, and that's been known since 1952 in the London experience. And increased risk can come solely from living near areas of high exposure to traffic, and that the effects of these multi-pollutants are further exacerbated by increased age. And increased age actually compromises physiologic functions, both of the cardiorespiratory and the hemostatic system. So there are many things that are going to affect this. So, as I said, both short and long-term exposures to PM 2.5 and PM 10 have been known to have negative effects on the respiratory and cardiovascular systems. Long-term exposure to PM in the elderly is associated with a prothrombotic state, so that means that these people are more likely to develop clots and those clots to form emboli, and they are at greater risk of myocardial infarction secondary to this problem. Long-term exposure to PM by proximity to heavily trafficked highways results in increased risk of deep vein thrombosis, and these are studies that have come out of Italy of people living near major highways. There's a very greater risk, particularly in males, but the study, while very interesting, didn't take into account the high rate of smoking, which is undoubtedly an additive factor in that population. We know that long-term exposure to PM 2.5 is linked to atherosclerosis. One of the most interesting studies from the Women's Health Initiative is that for every 10 micrograms per meter cubed increase in yearly exposure to 2.5, there's a 24% increase in cardiovascular events and a 76% increase in cardiovascular mortality. So clearly this is a pollutant that is very important to study. And the emerging data now links long-term exposure to PM with childhood respiratory disease, both with regard to asthma and diminished lung function, as well as increased vulnerability to chronic obstructive pulmonary disease in adulthood. And new research suggests that long-term exposure to PM 2.5 is associated with adverse birth outcomes, diabetes, altered neurologic development, and decreased cognitive function. So clearly this is an area requiring investigation. So what do we know from the animal models that have been studied thus far? Studies have largely been focused on young animals. The most typical animal that has been used is the ApoE negative mouse that's on a high-fat diet to develop an atherosclerotic model. There have been diabetic models. And there have been studies using the spontaneously hypertensive rat, but these studies have all been in young animals, animals that have not yet developed underlying cardiovascular disease. Studies have been conducted by concentrated air pollution, by intratracheal instillation, by insufflation. And ideally, what you'd like to do is to be able to mimic a situation where it's a normal inhalation of material in your environment. All of the studies that I mentioned have shown that pulmonary and hemostatic effects are present even in these young populations. And so our objective was to design a study, as Pat said, where to study defined pollutants and co-pollutants in an animal model that already has underlying cardiovascular disease. So this is how we've developed this model. We have studied in this model males. It will certainly be important in the future to study females. The animals are between 45 and 49 weeks of age, and we've used normal age-matched Wistar rats as the control population. These spontaneously hypertensive rats were originally derived from the Wistar strain, so we are, relatively speaking, within the same type of rat, if you will. We know that these SH spontaneously hypertensive rats have been previously used in PM studies, and as I said, the downside of those studies has been that they've been in younger animals. So while these animals, if they were maintained to the long term, would certainly develop underlying cardiovascular disease, they haven't done that early on. The other thing that's of great interest to us is that mature SH rats demonstrate spontaneous atrial arrhythmias and decreased heart rate variability. And this plays right into the studies that have been done and are very important when considering how the heart adapts to seeing a polluted environment. We worked with 4 different exposure conditions. Our control was filtered air. We worked with ultrafine PM, so UFPM at 250 micrograms per meter cubed, ozone at 1 part per million, and then the co-pollutants together. There was an hour, let me back up for 1 second. These animals were all instrumented so that we were able to study heart rate before, during, and after the exposures, and they were used in the study approximately 3 weeks post-instrumentation, so they had time to recover from the initial surgery. There was a 1 hour filtered air control period, a 6 hour followed by a 6 hour exposure, and an 8 hour filtered air recovery. And this was tightly controlled, and we thank Emily Wong, who's here with us today, for her yeoman's work in getting the exposures done and the humongous amount of the data that we're going to present today. The pollutant analysis was done from the DRI, Desert Research Institute, and as you can see in the paper that we've published, wasn't part of our CARB grant, but it turned out to be very important in considering how the co-exposure of ultrafine PM and ozone might actually alter the particles and have a synergistic effect. And as Pat said, for endpoints, we've looked at hemostatic, respiratory, and cardiovascular endpoints. So I'm going to start out by telling you a little bit about the white blood cell and platelet counts. Platelet counts from all these exposures were normal. There was no change in platelet counts. It didn't matter, but there was a very significant effect of white blood cells. So all these counts are done on a commercial hemocytometer. They're all standardized. And what's of great interest to us is that in both the normal Wistars and in the SH rats, exposure to the co-pollutants significantly decreased the white cell counts in the circulation. Now, normally, you would think with inflammation, this would be high. But the question became, and as you'll see in a few minutes, where did these white cells go? And the answer is to the tissue. So you're looking at 2 panels here. The normal Wistars are on the left, the SH rats are on the right. And this is looking at both filtered air on the top panel and ozone on the bottom panel. And I think you can see terminal bronchioles here, and you can see the alveoli. And in ozone, several things are immediately apparent. One is there's an increased cellular infiltrate. But in the SH rats, not only is there an increased cellular infiltrate, there is a lot of proteinaceous, my apologies, proteinaceous exudate present. And these rats are quite a bit more responsive than the normal Wistars. There's mural edema here, and there's both the normal Wistars and the SH rats. You can see a great deal of inflammatory cell infiltrate associated with the ozone exposure. These panels of UFPM and the co-pollutants show something really interesting. The UFPM animals really did not have big lesions. There were really minimal lesions, which is quite interesting. But both strains of animals exposed to the co-pollutants had really severe and really generalized lesions. So the terminal bronchiolar epithelium here and here was degenerative. It was necrotic. There was debris in the lumen. There were a lot of neutrophils and cellular infiltrate. And there was a great deal of edema present, particularly in the SH rats. You can see it here, associated with the terminal bronchiolar junction. And a lot of the alveolar ducts had a cellular infiltrate. And unfortunately, the magnification of this, despite the resolution of the image, isn't good enough for you to be able to see that. What you're looking at here are what you were looking at there are bronchiolar epithelium in the larger pulmonary airways. And you can see at the top in the filtered air, you can see the intact epithelium. You can see some of the connective tissue below it. And with UFPM, there are small areas of inflammation. There's an occasional neutrophil in the normal Wistars. And there are occasional disruptions of epithelium in the SH rats associated with UFPM. As soon as we look at the effects of ozone in these larger airways, it becomes apparent that this is quite different in the normal Wistars versus the SH rats. The overall organization of this epithelium in the normal Wistars has some small lesions, but the subcutaneous tissue, while slightly edematous, doesn't show much of an increased cellularity. However, when you look at the epithelium of the SH rats, there is definitely an infiltrate of neutrophils. There's disruption of this epithelium. There is edema, and there is loss of the underlying organization. This is really exacerbated when, and let me back up just a second. One of the things we do see with the UFPM, despite this small amount of disorganization, is the presence of thrombosis in microvessels. And we don't see that particularly anywhere else. There is a small thrombus here as well in the ozone SH animals. The exposure to co-pollutants results in a tremendous loss of the epithelium in the normal Wistars. You have cellular debris present in the lumen. There is edema present in the underlying tissue. And the situation is similar in the SH rats. You can see completely disrupted epithelium here. There's a large inflammatory cell infiltrate as well. So clearly there is a very large effect of the co-pollutants on these large pulmonary airways. We can go ahead and quantify this, and we chose to show the quantification specifically for ozone to make the point about how much more sensitive the SH rats are than the normal Wistar controls. Here we're looking at epithelial necrosis, so we showed you some loss of epithelium, and it's relatively small on a severity scale. In the normal Wistars, it's really substantial in the SH rats. It parallels the loss of ciliated cells, and interestingly enough, also parallels the presence of the exudate that I showed you that's present in the airways. There is, in both species, a considerable pulmonary neutrophilic infiltrate. And there is, and you can see the error bar here. You see this infiltrate in exposure to ozone in the normal Wistars, and it's significantly greater in the SH rats. So overall, this quantification tells us a great deal about what's going on, that there is small injury with UFPM, that there is significant infiltrate, leukocyte infiltrate with ozone, and that when we have those co-pollutants together, we have epithelial necrosis, loss of ciliated cells, the presence of exudate as well. So I told you that there was a loss of white cells from the peripheral circulation with exposure to the co-pollutants. And we'd like to tell you a little bit about what we think is happening from the hemostatic perspective. So one of the first things we did looking at these two groups of rats was to understand how reactive their platelets were. Because one thing we know about in older individuals is that platelets are more reactive the older you get. And so we looked at platelet reactivity with regard to exposure to the co-pollutants. And what you're looking at here is the expression of the major platelet integrin on the platelet surface, CD61, as a measure of activation. And one thing you can see is that CD61 increases in the normal Wistars and in the SH, but the only place in the normal Wistars that you see this increase that's statistically significant is with UFPM. And it's in those animals that we see the presence of microthrombi in the lung. The increase in the SH animals is seen both with UFPM and with the combined pollutants. So this is a situation where you're now in a prothrombotic environment, and that's the response of the animals. If we stimulate platelets on purpose, you see a very similar result. So not only are they responsive without stimulation by physiologic agonists, but when you add that, there is indeed greater expression of CD61 in the combined co-pollutants in both species of rats. So here you have a significant interaction where you have platelet microthrombi and upregulation of coagulation in the system that is priming the platelets to respond to agonist. One of the interesting things that platelets do when they become activated is that they shed platelet microparticles, and these microparticles are a prothrombotic marker. And the more microparticles you have, the more likely you are to develop a thrombus, and the more likely you are to develop an embolism. And you can define these microparticles in the flow cytometer by size. You actually have to use small little beads to do that. And so we looked at the presence of platelet microparticles associated with the exposures. And what we saw was really interesting. The place where platelet microparticles occurred was with ozone. And that's interesting because a long time ago when people were interested in preserving platelets for blood transfusions, they tried to ozonate them. And the idea being that you would use the ozone to kill bacteria and viruses. And indeed what they did was they produced many, many platelet microparticles, and those preparations were actually prothrombotic, and that was not a strategy that could be used. So the ozone sets up a prothrombotic environment in and of itself. So there are many places where the platelets are very activated. But it's really the co-pollutants where platelet activation is cleanest. There are three ways you can look at platelet activation. CD61, as I said, is on the surface of the platelets. And CD61 can either increase or decrease depending on activation. It could go either way. CD62P, or P-selectin, is an alpha-granule protein that is present on the surface of platelets, and it can be shed into the plasma. It is often used, soluble P-selectin can be used as a measure of activation. And CD63 is a lysosomal membrane protein that's only present on the surface and isn't shed. And what's interesting is the only place, the only exposure where you see a difference in these animals is in the presence of the co-pollutants. And here, the SH rats express a great deal more CD63, so they are more activated than the normal Wistars, and they shed both CD62P as well as CD61 on their surface. So again, another measure of activation. So we have increased platelet activation in platelets that were already primed to begin with, and the co-pollutants are what sets the system off. So what happens with the white cells and with white cell platelet aggregates? So you can look for neutrophils and monocytes using CD11b, leukocytes in general with CD18, and you can recognize platelets by aggregometry also as a large platelet aggregate set up in the size range the same as the white cells. And what you see here, which is very similar to everything else we've looked at, is that the changes are present in both the normal Wistars and in the SH rats with CD11b, only with the co-pollutants. And again, co-pollutants are an issue with CD18. So this is a decrease in expression of leukocytes generally in the peripheral blood. It corresponds exactly with the loss of white cells from the circulation. And the other thing we see now is that there's a diminishment of platelet aggregates in the peripheral blood. And this is accompanied by the presence of microthrombi, again, in the UFPM and in the co-pollutant exposures. And lastly, what we wanted to understand was, were there aggregates between platelets and neutrophils, which we know set up increased inflammation? And we looked at that by looking at CD61 and CD11b, and CD61 and CD18. And again, interestingly enough, careful what you touch, Dr. Schlegel. There is decreased aggregates in the UFPM animals, and there are decreased aggregates in the SH animals, and there are decreased aggregates of leukocytes, again, with the co-pollutants in the decreased animals. So all of this provides evidence that there is a prothrombotic, pro-inflammatory environment in the lung that is paralleled by what we see in the blood. So now we're going to turn this over to Dr. Schlegel, and he's going to talk to you about the cardiovascular system and all of the studies that we have done, and this is as was much of the previous work, all really done by soon-to-be Dr. Emily Wang, who's present here with us today.
All right, thank you. I think, am I working? Yeah.
Sounds like it.
Okay. All right, so moving to cardiovascular responses and looking at what we did as Fern mentioned, we instrumented these animals with telemeters and were able to measure heart rate and also EKGs throughout the exposure period as well as the recovery period. This is actually a trace from an experiment with ozone, and it shows what has been published before, especially for several years. With ozone, we know that there's an increase say a decrease in heart rate. This is the R to R interval. And so you could see that the R to R interval, the mean R to R interval going from the beginning, which is filtered air, beginning of exposure, and then there's a increase in the R to R interval, which means the distance between two beats is increasing, which means heart rate is decreasing. And this was demonstrated by Gary Hatch at EPA years ago as a means of looking at response in animals exposed to ozone. So we know this happens, and my background in looking at this in animal models has been the demonstration that pulmonary C fibers or bronchopulmonary C fibers play a very important role in inducing responses in rats. It induces rapid shallow breathing in rats, which is mediated via bronchial C fibers. C fibers are sensory nerves that innervate both the conducting airways as well as the terminal bronchioles in the rat. They send signals to the brain which induce these responses, which produces a inhibition of breathing, causing a rapid shallow breathing, actually, is what it's usually described as, and a increase in parasympathetic drive to the heart, okay, which is essentially an increase in tonic input to the heart from the parasympathetic system. Parasympathetic system, a little physiology here, slows the heart. Via the vagus nerve, the heart is innervated via acetylcholine-containing nerves, and the acetylcholine, when released at the heart, slows the heart, reduces heart rate, or increases the R to R interval, as it's shown here. The other thing that you can also see in this slide is that as we go from exposure or from filtered air before we start the exposure, through the exposure where our R to R intervals are increasing, and then to the end, that the variability is clearly, the band is much wider. What that really means is that our variability is increasing. So if we're starting to think about heart rate variability and how the variability of heart rate is changing, right off the bat, just looking at this trace, you can see that heart rate variability is increasing during exposure. So what we wanted to know, one, is how is heart rate variability changing? And the reason is because of this regulation of heart rate is dependent upon the two branches of the autonomic nervous system. The sympathetic system, which releases norepinephrine, induces increased contractility, increased heart rate, versus the parasympathetic system, which releases acetylcholine and induces a slowing of heart rate. And when the interaction between these two really determines the fluctuation and the variability in heart rate. So when we expose animals to a pollutant, and why do we study heart rate variability? Because we want to look at the change in autonomic input to the heart. And why that is important is because several studies have shown that with altered heart rate or autonomic regulation of the heart, you have an increased risk of developing arrhythmias and potentially essentially heart attacks or sudden cardiac death. So we want to look at heart rate variability for those reasons. So if we take this and you look at this trace, there's a couple ways to do this. Matter of fact, there's a lot of ways to look at heart rate variability, as we found out as we went through this process. And one way is just to say, okay, well, variability. How we look at any data set and look at variability? Well, we take the mean and you calculate the standard deviation. So that is essentially what is called time domain analysis of heart rate variability. What we do is we take a mean of the RR intervals, the distances between the R waves of the electrocardiogram, and take an average. We get a mean, and then we get a standard deviation. And that standard deviation essentially says that we have an increase in variability. So here, if you look at this for our exposures, our normal Wistars and our spontaneously hypertensive rats, you could see that what we have is, for the normal Wistars, is that there's, for the ultrafine particles by themselves, and for the combined pollutant exposure in the normal Wistar rats, there is a increase in heart rate variability. This is a measure of heart rate variability, which is essentially the standard deviation. You can see up there of NN or RR, you could even put that there. So it's the standard deviation of the R to R interval. And so you could see that indeed there's, as consistent with our plot, with ozone exposure, there wasn't much happening here. That trace was from a SHR rat, or a spontaneously hypertensive rat. In the normal Wistar rats, there's very little ozone effect, if any, but there's an effect of, in this animal, of, or these groups, of the UFPM and the combined exposure. Whereas if you look at the SHR, or the spontaneously hypertensive rat, there is a marked response, in increase in variability, with both the ozone by itself and with the UFPM. And so if you remember looking at the lesions associated in the lung that Fern just talked about with regard to the differences between the SHR rats and the normal Wistars, the SHR rats had much more or higher response to ozone with regard to the lesions in the lungs than compared to the normal Wistar rats. So here we're having much more injury in the lung associated with exposure, which you would assume that we're going to get much more activation of C fibers, and indeed with the ozone we get a much more marked change in variability. And that is carried over to the combined pollutant exposure. This is another marker of variability, and it just essentially shows the exact same thing. And again, time domain analysis. So this is another slide that kind of, we just want to illustrate when we go to something now called frequency domain analysis of heart rate variability, which is an attempt, or what we're trying to do here is pick out the role of the sympathetic nervous system and the parasympathetic nervous system. And the way this is done is by breaking the variability in the R to R interval into frequency domains or frequency components. Now, the classic way to do that is to use fast Fourier transforms to do that, but there's other ways to do that, which is what we're using, which is an autoregression approach, and I'm not going to go into the details of that. That would take way too much. But what's important about looking at these traces, and the top one is a spontaneously hypertensive rat exposed to ozone. And remember, we had a marked increase in variability in the time domain with ozone in these rats. And if you look at before the exposure started, you see that we can break up, and we look over at the parametric spectrum. You can see that the frequency domain can be broken into three distinct peaks. One is called the very low frequency, the next one is called the low frequency peak, and the next one is called the high frequency peak. The high frequency peak is usually considered to be a component that is driven primarily by the parasympathetic nervous system. Whereas the low frequency peak is a combination of both sympathetic, is affected by both sympathetic and parasympathetic interactions, and then the very low frequency is kind of off to itself, and it's kind of hard to understand what's actually happening with regard to that. Now, if we go down to the next slide, this is during exposure. At the bottom of the slide, same animal, but now during the exposure. And as you could see, if we look at the parametric spectrum again, there's a marked change. One of the things that we've done is we have lost that distinct peak in the high frequency spectrum. Now, as I said, animals that are exposed to ozone and sensitive to ozone develop rapid shallow breathing, which means that lung volumes are dropping during normal respiration. And part of this high-frequency peak is due to the activation of another group of receptors in the lung called slowly adapting receptors, and they're sensitive to increases in lung volume. So if you have an increase in lung volume above their threshold, they will be activated, and they produce a change in parasympathetic drive. But when we have ozone, you have rapid shallow breathing, you decrease the volume, and you lose that signal. So that's probably where we're working on our hypothesis, why we're losing that distinct high-frequency peak. But we also get an increase overall, and you have to look at the scales of these. That scale at the top is much smaller than the scale at the bottom. So our power is actually increasing overall. The actual area, if we take the area under the curve, the area under the curves at the bottom is actually higher just because of change in scale. And so that means we could look at this and say, well, we're losing that peak, but overall what you'll see is that there's an increase in high frequency power. So next slide. Yeah, that's right. I didn't do a Fern double jump there. So these are the actual analysis from animals, SHR rats that were exposed to ozone. And all I'm going to do today is present data on ozone-exposed spontaneously hypertensive rats, and I'll apologize for that. We are in the process of going through this considerable data set and actually finalizing our analysis. And because we had to reset some settings in our analysis, we have confidence in this data, and we're going to have to just wait to get everything else for all the other exposures. And again, Dr. Wong has done a considerable amount of work and continues to do a considerable amount of work on this in this area. So, but looking at this slide, you could see the very low frequency. You could see the dark bars are the exposure. There are very small white bars on there as well, and that is for the prior to or the filtered air exposure. So filtered air exposure, white bars, dark bars are the ozone exposed animals. And so if you start off at baseline or BL in the slide, you can see that there's really no difference between the animals that were exposed, which you would hope that were exposed to filtered air versus to ozone. Once we start the exposure, that changes. And we get marked increases in the power, again, the area under the curves, for very low power, and high frequency power, and overall total power. Now, what you can do, and what is often done to look at the balance between sympathetic and parasympathetic, is you take a ratio of the low frequency to the high frequency. And that will give you an idea of how the balance between low frequency and, I would say, sympathetic and parasympathetic nervous systems are interacting. Now, one of the things that is important here is that if you look at high frequency power, right, which is a measure of parasympathetic input to the heart, indeed, with exposure, it's going up, right, which is consistent with that activation of C fibers and the increase in tonic discharge of parasympathetic fibers at the heart, slowing the heart, okay? But also, if you look at low frequency power is going up more, right? And so as a result, when we look at the ratio, the overall ratio is increasing with exposure. So what that also means is that most likely sympathetic system is being affected, and we're getting an increased overall activity, both sympathetic and parasympathetic going to the heart, which induces a situation that is conducive to producing more and more severe cardiac arrhythmias, all right? And so if you could see now after exposure with recovery, the other two bars to the right of the large bars, this recovers after exposure. Okay, there's a period of recovery and things come back to baseline by the end of the exposure. The other important thing and more of a technical component of this is that we have a change in the waveforms, and this is important because of what we found out. The lion's share of work, like I said, was Emily's Long's tenacious work on this, and is that by using the autoregression approach that we do, we get more information out of this data. And the reason that is because the fast Fourier transform method fixes where the peaks are. In other words, it just assumes the peaks are there, they're stationary, they don't move. Whereas with the AR method, we're able to actually see how peaks move with exposure. And that means that when in studies that use fast Fourier transform, you're actually moving peaks from across these thresholds, which means that their data and their analysis is being influenced by essentially a dynamic that they can't identify using their technique. So what we'll get is from our final analysis with all these exposures is a better feel and understanding for how the autonomic nervous system is changing in its regulation of heart rate. So we have these marked changes with exposure, especially exposure to ozone. And as Fern showed, there's a marked pulmonary effect with ozone exposure in the spontaneously hypertensive rats. Now, if we go back and we take our EKG, which we've been collecting using our telemetry, and we have our blinded cardiologists sit down and look at them, what we see is that there's a whole array of arrhythmias that are associated with these exposures. One of the important things to note is that even our adult, mature, normal Wistar rats, remember how old these animals are, they're 45 to 49 weeks old, so close to a year old. Even the normal Wistar rats have spontaneous arrhythmias. And the spontaneously hypertensive rats also have arrhythmias. So now what we want to say is, how do the exposures actually change these arrhythmias? We have, as the slide demonstrates, atrial premature arrhythmias, atrioventricular blocks, so signal conduction blocks between the atria and the ventricles, and ventricular premature blocks, which are, or premature complexes, which usually are associated with ectopic events in the myocardium. So if you look at how exposure affects both of these, it's interesting that with regard to atrial events, especially premature complexes, the absolute number, this is numbers being shown here, number of events go up markedly in the normal Wistars in the particle by themselves. Marked effect. And then in the combined pollutant exposure, also, again, the normal Wistars actually have more events occurring. But there's also an increase in the SHR rats. Okay. Now, AV block, again, it kind of follows the pollutants as we combine them, so on and so forth. The UFPM, there's slight change, but not significant increases in both groups. Ozone, significant increase in both groups, but with the combined exposure, significant increase in both groups, but a marked increase in the spontaneously hypertensive rats. Look at the ventricular complexes, and again, we have this effect of a effect with the particles by themselves, both, and then an effect in the ozone where, again, if you look at the pattern here, especially with ozone and the particle by themselves, a greater effect in the Wistar rats as far as total number. If you look at the, then in the combined exposure, there's a marked increase in these in both groups, in both the normal Wistars and the spontaneously hypertensive rats. Now, if we sit down and we look at severity using a scale of severity, then what we see is that there's kind of balances what we saw out a little bit. So, and again, the severity of the arrhythmias is kind of balanced between where you have a marked increase with the particles themselves in both groups, a marked increase with combined pollutant with regard to ventricular ectopic beats, and supraventricular ectopic beats. You also have, again, an increase in both the overall in the normal Wistar animals, and then overall arrhythmias, the overall severity is increased primarily when you in both groups with the combined exposure. So along with these changes in heart rate variability, which we would expect, we're seeing changes in arrhythmias in both the total number of arrhythmias as well as their severity. Now, okay, that is very good. We're having increased arrhythmias. Arrhythmias by themselves are a severe cardiovascular event. Somebody who has having these types of arrhythmias going into a clinic, you know, they are immediately being treated and you know, being put on all kinds of medications and everything else. So by themselves, these arrhythmias are a marked cardiovascular event that could lead to sudden cardiovascular death. However, we also want to look at the myocardium to see whether this combination of thrombosis and prothrombotic state, the increased severity of arrhythmias and changes in heart rate variability led to any changes in the myocardium themselves. So these are 3 slides, and 3 different types of lesions were identified. I don't know whether you could see these all that well. Hopefully you can. In the first slide, there's focal organized necrosis, and you can see inside the little box here on that slide that there is an area of infiltrate and cells that are essentially a small area of organized necrosis. Right. And all these slides are from spontaneously hypertensive rats exposed to the combined pollutant. Okay, we're trying to adjust the lights a little bit here. There's also areas of what is called hypercontractility, and they're a little harder to see because essentially they're just yeah. With this stain are really indicated by these more pink, more eosinophilic regions or myocytes in the slide, indicated by the arrows. Now, it's interesting in that slide for hypercontractility down on the bottom hand right, there's also another area of focal organized necrosis. So we're seeing necrosis, we're seeing hypercontractility, and we're also seeing acute cellular necrosis. So if you look at the far hand right there, and you look at where the arrows are, and you look at those myocytes that have the arrows going to them, you can actually see vacuoles in those myocytes. In other words, those cells are dying, in the process of dying. They're breaking up, they're in the process of dying, and at some point would probably lead to infiltration of cells and probably lead to our focal organized necrosis. So what we're seeing in the spontaneously hypertensive rats, which we do not see in the normal Wistars, is a significant increase in necrotic and hypercontractile events in the myocardium of these animals. And they are present with PM by itself, or the ultrafine PM itself, with with the combined pollutant. So both for acute cellular necrosis and for the hypercontractility. Whereas overall, we had increased for the focal organized necrosis, there's an increase in every exposure group for the spontaneously hypertensive rats. So we have our events of thrombosis, we have our events of heart rate variability changes, our changes in arrhythmias, but we're looking at two groups of animals. A group of animals that have, yes, some degree of, because of their age, compromised cardiovascular function, whereas another group of animals, because of their genetic makeup, develop spontaneously hypertension at about 20 weeks of age, and then continue to develop, because of that hypertension, marked lesions in their heart. Their hearts become fibrotic, hypertrophied, and there's marked effects on the myocardium because of their hypertension, as you would expect to see in a hypertensive individual as well. And so in those individuals, because the myocardium is already compromised, when we put all these other events on top of it, we actually are inducing cardiac lesions. And that's really important. And I say, it's actually really amazing that we didn't lose any animals in this study. And we actually have one animal that the cardiologist looked at the EKG and was amazed, because essentially the animal's heart stopped for like a minute or 2 minutes of time, and then all of a sudden he started to beat again. Came back to death. So the Lazarus rat. It was quite amazing that these animals didn't have, we didn't lose any of them during the exposure. So anyway, that kind of wraps it up. We have rats with underlying cardiovascular disease that are clearly susceptible to injury of the myocardium, but also, as Fern demonstrated, they are more sensitive to the oxidant stress of exposure. So that sets them up as well. So more sensitive to oxidant stress, which induces a greater response, especially in autonomic effects on the heart, and that contributes to the arrhythmic effects as well. Both strains were similarly affected by the combined pollutants if you look at it across the board, but when you get down to the tissue level and you look at lesions in the myocardium, SH rats that are demonstrating that effect. So the overall effects of this exposure is that the combined exposure had a greater biological potency overall, and with regard to all of the exposure groups in both the normal Wistars and the spontaneously hypertensive rats. And so overall, what we see is effects on hemostatic events, so prothrombotic events, changes in heart rate variability, changes in arrhythmias and severity of arrhythmias, and an effect, especially in the spontaneously hypertensive rats, in the effects in the myocardium with regard to actual lesions and necrotic lesions specifically. One of the things that we're going to be doing and continuing on, and which we now continue to have funding from California Area Resources Board, will be to better characterize those lesions in the myocardiums of these rats, and to look for whether these actually are, those lesions are due to ischemic events, which would be very similar to a heart attack. Are these rats having small heart attacks during exposure? So that will be the next phase of this research. So thank you, and have any questions?
Okay, yeah. Okay, I'd like to thank speakers, both Fern and Ed today, and like to know, open it up for any questions. Do we have any questions from the audience in here? Let's see, we have any questions online. Well, I guess I do have a question. It does kind of relate to the follow-up project, which we just got funded, so we look forward to the results from that. What exactly are the mechanisms you're looking for to get from the, I guess, the cardiovascular, the heart rate variability, arrhythmias, to the necrosis? You mentioned ischemia. Were there any other, or is that the main one?
The primary one is ischemia. We also want to, because that's really a question. When you look at those lesions and you have a question about what's inducing those lesions, the typical event that causes necrosis in individuals is ischemic lesions, a clot, a thrombi developing, or secondary to a block of an artery due to, what do you call it? Cardiovascular disease, or atherosclerotic lesions producing plaques and clots. My son, the cardiologist, would kick me in the head for mispronouncing that. But that would be consistent with, again, it's Emily, would be consistent with ischemic events. And so it's really important to see whether that's what we're inducing. Are we inducing ischemic events? Unfortunately, in the first go-around, we did not use a marker of ischemia. In the funded research, we will be looking at a marker, connexin 43, which is a proven marker of ischemia in the myocardium. So we'll be able to associate the lesions with ischemia.
Okay, thanks. One thing which I know you talked about it and we had an introduction, this was a very short study in terms of you have an acute exposure, a recovery period, then you saw these effects, and it's kind of hard to know what would happen. Some of the effects you're seeing, would those be permanent, or would you expect them to recover? I'm just curious what would happen if that longer term.
I think with regard to looking at, especially the spontaneously hypertensive rats, those lesions in the myocardium are permanent.
Okay.
I mean, it's like having a small heart attack, and I think that's a really good way to look at that. Those are many cardiac events that are occurring, and the damage to the myocardium is not going to repair. It's going to become fibrotic and essentially loss of cardiac function over time.
Yeah, I'd be interested to see if you could wait a while after recovery and see if they're more sensitive to another exposure later on. Yeah. Okay, do we have any questions from the room or any online? No, I guess no more questions. So again, I'd like to thank our speakers, Fern and Ed, today for an excellent talk. And if you have any additional questions, you could send them by email and I could forward those to the speakers. Thank you.